Okay, guys, welcome to Let's Get to the Marks, going through AQA AS Biology Paper 1. This is the 2018 paper. There's 10 questions, hour and a half. I recommend that you pause the video at the start of each question, grab a pen, grab a piece of paper, have a go at the question, make your revision active. You won't learn best with passive learning. Make your revision active, have a go at the question, then unpause the video and watch me go through the explanation. All right, let's go. Question 1.1, figure 1 shows part of the blood circulation in a mammal. God, I forgot how to speak there. We can see we've got the lungs in a rectangle here. We've got these various arrows connecting the different boxes together. These arrows represent different blood vessels, and these rectangles with labels obviously represent organs and parts of organs. So use figure 1 to give the letter that represents each of the blood vessels. So we need to, what letter is the aorta? What letter is the renal vein? What letter is the vena cava? So I'm going to go through each and explain it. So, which one of these is the aorta? Now, the aorta goes out of the heart and takes oxygenated blood around the body. So we're really looking at D here. You can see that is the blood vessel that's leaving the heart. Arteries go away. Think of the A for away. They leave the heart and they take oxygen around the body. So we can see D must be the aorta, as the other arrows are going to the heart, and this one going to the lungs is the pulmonary artery. So let's look. D is the aorta. What about the renal vein? So the renal vein, anything with the word renal, is to do with your kidneys. Now, veins go towards the heart and carry deoxygenated blood. So E would be like the renal artery because it's coming off of the aorta. It's carrying oxygenated blood to the kidneys and supplying them with oxygen for respiration. And G would be the renal vein because veins bring blood back to the heart, deoxygenated blood back through the vena cava. So we're looking at the answers D followed by E, and then let's check out that last one, the vena cava. I just mentioned it. The vena cava is the big vein that collects all the other veins together and brings deoxygenated blood back into the right atrium. So that is going to be F. So your answers here, folks, hopefully you get all three of these marks right, should be D, G, and F. Lovely jubbly. 1.2, name the blood vessels that carry blood to the heart muscle. So this is very specific. And what we're going on about here, let me just do a little bit of an explanation for you. So as the aorta D leaves the heart very quickly, there is a little artery that splits off and goes back to feed all of the muscle walls in the right atrium ventricle and the left atria and left ventricle so that they can contract. So it's providing oxygen to the muscles. And of course, what do we call this blood vessel here that peels off of the aorta? Hopefully, you know, it's the coronary artery. Okay, and you have to be very specific there. They're not going to just accept you saying artery. It's got to be the coronary artery. Wunderbar. Um, yeah, there we go. Or we can say the coronary arteries as they split up into lots of different little arteries that deliver oxygenated blood to different parts of the heart muscle. Okay, let's zing on. We've done 4 marks so far out of 100. We don't want to take this long. 1.3, figure 2 shows a photograph of part of a mitochondrion from a mouse liver cell taken using a transmission electron microscope at times 62,800 magnification. That's a really big mag. Produce a scientific drawing of the mitochondrion in figure 2 in the box below. Label the following parts of the mitochondrion on your drawing. So hopefully you remember from your GCSE biology, if you did GCSE biology, there are certain rules when you draw this. So it means we mustn't have any unbroken lines. It's got to look pretty similar. We need the magnification written underneath. We need to label it correctly and nothing shaded. No crossed lines, no shading. So this is my crusty drawing. Now, I shouldn't be drawing in red, but I selected it earlier, so I'm just going to stick with that. So look, this, oh my gosh, straight away there, I lose a mark. So there we go, right? Doesn't have to be perfect, but it should be recognizable. Look, my lines are not crossing, okay? So no sketching or anything like that. There we go. Really and truly, I'm going to do this in black because that in red just looks awful. Okay, so let's zing I'm going to do that. And then it's really the inner cristae that we want to pay attention to. So the cristae, we want to do something like that. Okay, you get the idea. And then it's got to look similar. Oh my days. Did you see that fat one there? No, did he? That's pretty grim. Right. Let's go up there. I did get a level 4 in GCSE art, although in my day it wasn't called a level 4. It was a D grade. So I don't even know if a D is a level 4. Maybe a D is actually a level whatever. Okay, what do I need to do? Right, so I've got no crossing, no shading. You've got to write the magnification when you're doing cell drawing. So 62,800. And then you need to label. So we need to label two things here. They want us to label the matrix and the cristae. The matrix, folks, um, is the kind of cytoplasm of the mitochondria. Um, and we also have cristae like that. Now, you might be asked about what these circles are in here. They could be glycogen or they could be other things, enzymes or such. Right, let's write in cristae. So this is the cristae, the finger-like structures. Okay, and that will get you 4 marks. So one for correctly labeling the matrix and the cristae, one for not having any crossed lines, broken lines, etc. No shading. So one for that, and then one for the mag. Great. Let's zing zing to the last part of the question. Oh, that is the last part of the question. Okay, we're on question 2.1. So, what is a monomer? It's worth 1 mark. What I reckon you do is you pause the video, have a go at this 2.1 and 2.2, write it down on some scrap paper with a pencil or a pen, and then check the answers and my explanation. Okay, here we go. What is a monomer? So, a monomer is basically a small molecule that makes up a polymer. So, that would be my answer. A small molecule that can be joined together. You can say makes up um, to form a polymer. It's a repeating unit. You could call it a repeating unit, repeating units of a polymer. Um, or you could say small molecules can be joined together by condensation reactions to form a polymer. Again, we don't need all those terms really. They just want to get through this, but it's always good fun to write them all down. Right, that's 1 mark. Right, now 2.2. Lactulose is a disaccharide. What does disaccharide mean? You've got to ask yourself. Formed from one molecule of galactose and one molecule of fructose, other than both being disaccharides, give one similarity and one difference between the structures of lactulose and lactose. Right. On your specification, you don't have to know about lactulose, but guess what? They've told you about it. And this is called an application question. So you're using your knowledge, your content that you have learned off by heart, and then you're applying that knowledge to discuss the structure of lactulose. And we want the similarity between lactulose and lactose. And you should know, what do they both contains? So both contain, oh my good gosh, both contain glycosidic bonds. So both of them have a 1 to 4 glycosidic bond joining them together. They're both disaccharides, but they told us that there, so we can't say they're both disaccharides. Now we want a difference. They both contain carbon, hydrogen, and oxygen, I know, but they want the glycosidic bonds for the mark scheme. So what is one difference? Well, this is where your content comes in. You should know the structure of lactose. You don't have to know the structure of lactulose because they've told you it's a galactose and a fructose. But actually, we would say lactose, and you have to know this off by heart, is made, whoa, moed is made from a glucose molecule and a galactose molecule. So lactose is actually made from that, and that's what you have to know. And if you want to get really funky, you could just, I mean, they've already given us this in the question, but I'll just put it there. So, whereas lactulose is made from what does it say? Galactose. Oh my good gosh. Um, oh gosh, this is all going so pear-shaped. There we go. I mean, a similarity, therefore, I would say a similarity in the structure, therefore, that we could add up here is that both contain galactose, right? So I would add that as that should be a similarity. So other than both being disaccharides, give one similarity. So I reckon both contain galactose, whereas lactulose has fructose and lactose has glucose instead of fructose. So yeah, that would be another similarity there. Okay, let's move on. I'm whiffle waffling. Oh, what the beep? Right, 2.3. Following the digestion and absorption of food, the undigested remains are processed to form feces. Nice. In parts of the intestine below the ileum. Feces of people with constipation are dry and hard. Very nice. Constipation can be treated by drinking lactulose. Lactulose is soluble, but is not digested or absorbed in the human intestine. Use your knowledge of water potential to suggest why lactulose can be used to help people suffering from constipation. Again, this is an application. Not only do you need the knowledge, you need to apply it to a different situation. So what we've got going on here, if this is the lumen of your intestine, what they're saying is that the lactulose, which I'll use an X to show lactulose, the lactulose cannot go out. It can't do that, right? It can't leave the intestine because it's not soluble. So we've got our cells surrounding the intestine and on either side. And what they're saying is this lowers the water potential. So what do you think is going to happen? Well, water is going to move in by osmosis from a high water potential to a low water potential because the lactulose has lowered the water potential in the lumen. And that means water will move in by osmosis from the surrounding cells into the feces or into the lumen and mix with the undigested food. And it will soften the feces, soften the constipated poo. Lovely. Okay, let's put that answer down. So we want to say lactulose lowers the water potential in the lumen of the small intestine. Water moves in from surrounding tissues down a water potential gradient. So from the epithelial cells, etc., moves in down a water potential gradient by osmosis. This softens the feces and relieves constipation. So essentially, that's how that works. If you're constipated, take some lactulose. It will lower the water potential inside the lumen of your small intestine, and water from the cells around the intestine will therefore move in by osmosis and soften the feces. Great. Okay, let's move on. Now we've got 2.4, the last part to question 2. Lactulose can be used to treat people who have a too high concentration of hydrogen ions in their blood. A normal range for hydrogen ion concentration is 3.55 times 10 to the minus 8 to 4.47 times 10 to the minus 8. So that's the range of the normal H ion concentration. A patient was found to have a blood concentration of 2.82 times 10 to the minus 7. Calculate the minimum percentage decrease required to bring the patient's blood concentration into the normal range. So their hydrogen ion concentration is larger. Look, it's to the minus 7. The range is to the minus 8, so it's smaller. What is the percentage of decrease that we need to do to bring it within the minimum range? Our minimum range is, well, our range is 3.55 times 10 to the minus 8. And this range stretches all the way down to 4.47 times 10 to the minus 8. So, yeah. Now, what do we want to do? We want to try and get it into the minimum range. And so in order to get it into the minimum range, we just have to bring this number, which is 2.82 times 10 to the minus 7. We only just have to get it into the minimum range. So it doesn't have to go in here. We just have to use this number. So what we want to do is work out the difference between these two numbers. This is the original of our patient. This is the original H ion conc of the patient. That's what's their hydrogen ion concentration. We need to get their H ion conc um, here. We need to get it there. So we're going to find the difference and divide it by the original and times 100. So let's go ahead, smash that in the calculator and see what we get. So 2.82, put the big number in first, times 10 to the minus 7. I know it doesn't seem like a big number with a minus 7, 7 zeros in front, but that one's got 8 zeros in front. So now we're going to take away 4.47 times 10 to the minus 8. And we're going to get, this is our answer. We're going to get 2.373 times 10 to the minus 7. And I would say that is going to get your first of the 2 marks. And now what we're going to do, that's the difference between the patient's actual hydrogen ion concentration and the bit that's closest to them in the range that we can nip it into. So now we're going to divide this by the original, because when you're doing percentage decrease, you do the difference over the original. So the difference is 2.373 times 10 to the minus 7. We're doing it over the original, and then we're going to times 100 and find out exactly how much it had to change. Times 100, and that is going to give us 84.14%. Now, I would just do 3 significant figures, so I'm going to just switch to black because that's not showing up very well, is it? And I would do 84.1%. There we go. Beautiful. And that's going to get us the second mark. All right, that's the end of question 2. Let's zing zang to question 3. Draw and label a single DNA nucleotide. So why don't you grab your pen, grab a bit of paper, pause it up, see if you can scribble out a nucleotide and label it. Go. All right, let's go through it. First of all, we have the phosphate group. Okay, I'm just going to label mine P. You would need to do arrows and do the full naming. You cannot just label it with letters. I wouldn't take the chance that you can. And then we've got a pentagon and a rectangle. So circle, pentagon, rectangle. We've got these lovely bonds. So this is my deoxyribose sugar. So we should definitely say that it's a pentose sugar because it contains 5 carbons, but it's a deoxyribose. And over here I have my nitrogenous base. Now I could do any of these, so I might label that like adenine or whatever. So let's write this out. So we got phosphate, we got deoxyribose sugar, and then we've got nitrogenous base. And yeah, I've gone for A, but I could have gone T, G, or C. You wouldn't get a mark if you put U because U is in uracil and is not a DNA nucleotide. So make sure you don't mess that up. Right, give 2 features of DNA. So 2 features of DNA and explain how each one is important in the semi-conservative replication of DNA. Okay, so let's think about our DNA. What do we know about DNA? Well, DNA we know is double-stranded. We know it's got complementary base pairing. We know that allows hydrogen bonds to form between the bases on either adjacent DNA strand. Okay, so let's go through, name a feature and explain. So let's start with it has hydrogen bonding. Hydrogen binding. I want to say it has hydrogen bonding. And what is important with that, it is easily broken or unzipped. Now, why is that? Well, hydrogen bonds are actually the strongest of our intermolecular forces, but they're weak compared to ionic bonds and covalent bonds. So those disulfide bridges, these bonds here, for example, these are covalent bonds. They're a lot stronger than hydrogen bonds. So having bonds that are easy to break, where you don't break other bonds, is important. So a hydrogen bond's easy to break, so the DNA can unzip. What else can we say? Oh, it is double stranded. Of course, it's got a double helix. We could talk about the sugar phosphate backbone protecting the DNA and reducing the chances of mutation. But it specifically wants us to talk about it in terms of semi-conservative replication. So I'm going to say it's double-stranded. Each polynucleotide strand, let's just say each strand can act as a template during replication. And that sounds pretty good. That'll get us 2 marks. Lovely jubbly. We could also talk about complementary base pairing. Give an example: adenine to thymine or guanine to cytosine. This means that the replication is very accurate or very specific, and allows the replication to be accurate because it will the complementary base pairing of the DNA nucleotides, the free DNA nucleotides, will only join to specific bases. Okay, let's move on. So we're now looking at 3.3, replication of mitochondrial DNA, in brackets mtDNA, MT stands for mitochondrial, is different than that of nuclear DNA. The replication of the second strand of mtDNA only starts after 2/3 of the first strand of mtDNA has been copied. Okay, so replication of the second strand only starts after the first strand has been 2/3 copied. A piece of mitochondrial DNA is 16,500 base pairs long. It's replicated at a rate of 50 nucleotides per second. Tick the box that shows how long it would take to copy this mtDNA. Here's our DNA, right? Let's just say there are 2 strands, right? I know mitochondrial DNA is not going to look like this. It's more of a loop. But let's just look at the 2 strands of DNA. It says, how long does it take? So we're going to go, there's 16,500 base pairs. So to copy one strand, there are 16,500 complementary base joining points. And 50 nucleotides join a second. So I'm going to do 16,500 divided by 50. It's going to take 330 seconds for one of the strands to be copied. Now, if they both started getting copied at the same time, then the total time would be 330 seconds. But what they're saying is that 2/3 of the way down here, what's 2/3 of 330, peeps? 2/3 of 330. So let's do, what's that? 0.66 times 330, roughly. Okay, that's not very good, but yeah, it's 220. So once we get to 220 seconds, bearing in mind that it takes 330 seconds to copy the strand, so once we get there, 220 seconds down, this second strand starts to be copied. So what's the total amount of time it's going to take? Well, we've already got 220 on the board. And then in 110 more seconds, it's going to be there and it's going to have completed that, and then another 220. So it's going to be basically 220 add to 330, because this whole strand is going to take 330, but it doesn't start to be and this is going to be the limiting factor, the one that takes longer, the second strand, but it doesn't start to be copied until this one is 220 down, and then that starts to be copied. So we just want to go 220 add 330, and the answer is 550. And if you can make sense of what I was waffling on about there, 500 points. Well done. Okay, let's get on to question 4. So figure 3 shows part of a prokaryotic cell. Here's figure 3. Have a little look. We got the cytoplasm, and then we have W, X, Y, and Z. Name structures labeled W to Z in figure 3, and it's worth 2 marks. Why don't you grab some paper and a pen? Press pause on the video now. Have a go. All right, I'm going to go through the answers. So Where do we want to start? W. Well, what is always touching the cytoplasm? You got it. It's the cell membrane. So hopefully you went for that. Cell surface membrane, membrane, or just cell membrane. Yeah, you get the idea. We're not going to call it phospholipid bilayer, but we will call it cell membrane. Okay, X. What is surrounding the cell membrane? Well, that's going to be the cell wall. And we need to be a little bit specific because we're in A level. So we need to say what substance that's made of. And so what is the cell wall of a bacteria or a prokaryote made of? It's not cellulose. That's found in plant cells. It's not chitin or chitin, as people like to call it. That's found in fungi. What is it in the cell wall of prokaryotes? Well, there's three possible answers here. You can say murine, you can say peptidoglycan, or you can just call it a glycoprotein, which is a more vague answer. It's not the specific name, but essentially they have a protein with a sugar attached, and the specific name, peptidoglycan or murium. Okay, and that will get you that mark there. Now, why? There is a mucus layer that lies on top of the cell wall of bacteria. Bacteria are slimy. If you ever felt like chicken or meat when it starts to go off, it gets really slimy. What is that? Mucus or that slime layer that's around, it's not really mucus, but it's around the bacteria. Hopefully you know what it's called. It's called a capsule. Now I reckon there's a big misconception here and quite a few people will get this wrong and put capsid. Let's not forget the word capsid is found in viruses. Capsule is the slime layer on bacteria. Okay, and finally, Z, we've got the tails. Again, people might make a mistake here because bacteria have pili, which come out all around, but this is the flagella or flagellum. As we're pointing to one, I'm just going to call it flagellum. And this is like the tail that moves in a corkscrew fashion and powers the bacteria through fluids. Okay, there we go. How did you do? It's only worth 2 marks. I know I put 4 ticks, but there we go. Let's move on. Right, let's look at 4.2. Pause the video. Name the main biological molecule in W and X. Oh, I kind of already did that. I already did that for the main biological molecule in a cell membrane. You know what that is, guys. That's a phospholipid. And I already did the second one. Got a bit excited. You could just put cell wall for X. That is the peptidoglycan, or you can call it glycoprotein, or you can call it murium. So there we go. You didn't have to put all them funky names for cell wall, but I thought, why not? Hey, there we go. That's the chemical that's in the cell wall of prokaryotic cells, which is generally bacteria. Right, 4.3. Name the process by which prokaryotic cells divide. You might remember this actually came up in GCSE as well. They don't divide by mitosis, they do binary fission. So hopefully you got that right. And now we have a disgusting little mathsy question. So let's have a read. Some prokaryotic cells can divide every 30 minutes. A liquid culture contained a starting population of 1.35 times 10 to the 4. What's that? 13,500? Something like that. Assuming each cell divides every 30 minutes, calculate how many cells there will be in 3 hours. Assume no cells die during this time. So, first of all, I want to know how many divisions, how many doubling is there going to be? So it says they divide every 30 minutes, so every 3 hours. So I want to go 3 times 60. Oh, by the way, why don't you pause the video and have a little go at this one? This one I think is slightly easier than the normal one. So we've got 180 minutes, but they divide every 30 minutes. So 180 over 30 is 6. We have a total of 6 divisions. So we want to multiply it by 6 times. So we want to do 1.35 times 10 to the 4, and we want to double that 6 times, right? So we want to basically do 1.35 times 10. Now, some people will mash this up because what they'll do here is they'll go, oh, 6 times 2 times 12. So they'll just times it by 12, thinking they can do that. No, you've actually got to do times 2 times 2 times 2 times 2 times 2 times 2. Oh my good gosh, let's smash that in the old calcs and see what we get, eh? 1.35 times 10 to the 4 is 1.35 times 10 to the 4. 1.35 times 2 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 Now, did I do that six times? One, two, three, four, five, six. I did. And you're going to get 800. Oh my gosh, I can't read my calculator. My brain's gone. 864,000. Now, does it want, now you could write that like, in standard form if you wanted. I'm just going to leave it like that, but you can put it in standard form as they gave us the population of cells in standard form. So you could turn that into standard form. What would it be? 8.64. Okay. What was that times 8.64 times 10 to the 5? Yeah, so something like that. Anywho, I'll leave it like that. That way, I'm less likely to make a mistake when I try and convert it into standard form. There we go. That is the end of question 4. Let's zing to question 5. 5. Figure 4 represents a triglyceride. Have a little look. P and Q. We've got the bonds in between. Name the molecules represented in the figure by box P and Q. Name the bond between P and Q. This is worth 3 marks. All right, let me just shrink that down there so we can fit it all on the page. Pause, scribble it down, have a go. Here we go. Answers coming. Let's go. Right, box P, that's going to be glycerol. And again, this is kind of GCSE knowledge. You know that lipids are made of glycerol and 3 fatty acids. So we've got fatty acids. Okay, and that's going to give us 2 lovely jubbly marks. Glycerol and 3 fatty acids makes a triglyceride. Triglycerides are great for insulation, great for energy storage. Name the type of bond between P and Q. Hopefully you know the name of the bond. It's an ester bond, not a phosphodiester bond, which is most likely the most common mistake. If you put that there, you're halfway there. But phosphodiester bonds are found in nucleotides or between nucleotide monomers within a polynucleotide like RNA or DNA. Okay, let's move on. Right, we're on to 5.3. Describe how you would test a liquid sample. So they've already mashed it up and blended it. So we don't need to say mash it up and blend it because it says liquid sample for the presence of lipid and how you'd recognize a positive result. And that's 2 marks. There's a whole number of ways to test for lipids. Why don't you see if you can get 2 marks for the lipid test? All right, here's what I would do. I would shake with ethanol and then I would pour into cold water and you get an emulsion. So it's called the emulsion test. Um, a positive result, it goes, or it forms would be better. It forms a milky emulsion, like a white cloudy emulsion. Okay, cool. Or precipitate. There we go, there we go, there we go. It's known as the emulsion test. Shake with ethanol, pour in cold water, it goes milky white. Lovely. I know there's the Sudan 3 test with the red layer. There's you can use like certain type of paper and rub it and see if it goes transparent. I'd have to check the mark scheme to see if they accept that. They should accept any um proper test, and it doesn't say what the test doesn't hint that the test has to be a specific type. All right, guys. Oh, it's the end of question 5. Whoa, we're halfway there. Yes, I didn't take music and I got a D in art, which is like a level 3, so you can probably guess. All right, let's move on. Let's move on. Figure 5 represents the phylogenetic classification of 4 different species of fruit fly. Back on topic, back on topic. Figure 5 shows a hierarchy. Explain how. 2 marks. Go on, have a pause, have a go. Or I'm going to go through it. So we've got Drosophilidae, then we have Zaprionus, Neglectus, and Sexvitus. Drosophila erecta, excuse me, Willistoni. Erecta Willistoni. Okay, that's yeah, interesting. So how does this show a hierarchy? So you're talking about your king prawn curry or fat greasy sausages, basically. How does this show that there's a hierarchy? So what it shows is that we have some large groups, and inside the large groups are smaller groups. For instance, we could have the kingdom of animals, and then within animals, we have smaller groups like mammals, like fish. You get the idea, right? So how it shows that we have a hierarchy, we can say we have large groups with smaller groups inside them. But also, how it shows a hierarchy is there is no overlap between groups. Look, The Sexvitus and the Neglectus, that's happened to me a lot. These two groups do not overlap, or the Erecta Willistoni, yeah, that doesn't overlap either. You can see they're separate, all the groups, and the Zaprionis Drosophila do not overlap each other. So what we can say is there is no overlap between groups. So officially, you're not a fish and a bird, right? There's no overlap between birds and fish. You get the idea, or humans and monkeys, or anything like that. So when you put them in a classification system with a hierarchy, we don't have any overlap between the different groups. Anyway, I'm waffling. So we want to name the taxon to which Drosophilidae belongs. So that means like the level to which Drosophilidae is. So what we want to do is we want to say our little, this could be quite difficult, but we want to say our little rhyme in our head and go kingdom, king prawn curry, or fat, greasy sausages. I mean, you can add domains to the top of that if you like with the domains. You can say delicious king prawn curry or fat, greasy sausages. But essentially, let's work our way back. These both begin, check it out. These ones all begin with a lowercase, erecta, Willistoni, Sexvitus, and Neglectus. What one begins with a lowercase? So these are the species. What's next? Then we have these ones with the capitals in front. These must be the genus, species. We've ticked off genus. We have Drosophilidae left, and that must be the family. And this diagram is not showing us the order, the class, the phylum, or the kingdom. So I would say that Drosophilidae is the family. Okay, family, there we go. Bosh. Let's go down. Drosophila fruit flies display courtship behavior. One of the stages of courtship is singing by males. If you heard my singing earlier, I would not do well as a fruit fly. I'd definitely not have kids. Normally, a male will produce a sign song in which the continual noise is made, and a pulse song in which there is a continual noise with some louder peaks. Scientists showed fruit flies a visual stimulus that made them sing. They made recordings of these songs. Figure six, Sean Connery, shows the recordings of the songs of three fruit flies over the same period. So we've got the male D erecta, here's its sign song, and then it goes into a bit of a pulse song. And then we got the D Willistoni, here's its sign song, and then a pulse song. And then we got the female D Willistoni, and we got the sign song and a little bit of a pulse song. Now, what are they actually going to ask us? D erecta and D Willistoni are closely related, but different species, i.e., the kind of fertile offspring. They can't reproduce to produce fertile offspring. Describe evidence from Figure 6 that supports this. So if we look at D erecta and D Willistoni, what evidence supports this, that they're closely related? So check out the sign song. The Willistoni male and the erecta male have roughly the same length sign song, and the female Willistoni roughly same length sign song. Maybe that's a little bit short, but definitely the yeah, it's a little bit longer. Roughly the same length sign song for the erecta and the Willistoni. And then it asked us to point, and are they the same amplitude? They're roughly the same amplitude as well, which means the same height of the wave. However, when we get onto the pulse song, the difference is the erecta had like 1, 2, 3, 4, 5 large peaks. So a different number and different position of the peaks compared to the Willistoni, which has a different number of peaks. I can only count 3 big peaks there and a different spacing between the peaks. So there you go. There's your similarities and your differences. Let's go down and put that in the answer. So what we want to say here, what evidence supports it? Sign song is the same length. I'm not going to bother writing the full thing out because I'm getting lazy now. Is the same length between the species. And I probably shouldn't say it's the same length. It's not exactly the same length. So I should say, let's clarify that. Otherwise, some naughty examiner might not give me the mark. Roughly the same length or similar length between the species. However, the difference is the pulse song is different. Now, we can't just say it's different. We have to say why. The large peaks appear in different places along the length of pulse song, and um, each species has a different number of large peaks in the pulse song. Oh my good gosh. There we go. 6.4. Scientists repeated the experiments using female fruit flies as the visual stimulus. When a male and female D. Willistoni were together, their songs led to mating. So they showed them, they used female fruit flies to basically get them excited. When a male and female D. Willistoni were together, their songs led to mating. So they sung and then they mated. When two female D. Willistoni were together, their songs did not lead to any attempt to mate. Use information from figure 6 to suggest why the two females did not attempt to mate. So why is it that the two women didn't get it down? Their songs are obviously going to be the same. But so what we can suggest here is in order for them to understand, hey, am I having sexual intercourse with a male or a female? Um, they go by the song. So we can see here that the large peaks appear in different places, the amplitudes, the length of the sine wave. We can see here, look, the male Willistonia has a longer sine wave length. The position of the large peaks in the pulse song appear at different places. So they can recognize that the females can recognize the male is different enough that it's not the same gender, essentially. And we need to just construct that into an answer using the evidence with the length and the positioning of the pulses in the pulse song. So we want to say the female songs are identical. Um, the male song has peaks in the pulse at different places. I'm sure I could make this more specific. I could say different large peaks appear in the pulse at different places between male and female. That's what I'm getting at. But I don't want to rewrite my whole answer. Okay, and then we might want to say the length of the sign is different in male and female. Um, the females cannot recognize a male song as no males are there. So they're listening out and the other one is singing at them. They're going, no, you're female, you're not a male. I'm not getting it down. Basically, the females cannot recognize a male song as no males are there. So no mating occurs. So that's why they don't even bother attempting to mate. So if you were a female and you wanted to trick another female into mating, but what would be the evil? You could mimic the male song, I guess. But what would be the evolutionary benefit to that for the birds? I don't know, but it's probably some interesting research on animals there that you could do. Probably, I wonder if there is some species that actually do that. They mimic different genders and stuff, and to what advantage that would be. That would be an interesting topic. Right, question 7 and up, Wiffle Waffle. What is the proteome of a cell? So you should know this off by heart what the proteome is. So it's a nice little memory recall one. It is, oh my days, it is the full range of proteins coded for by the DNA in an organism. Is protein I before E or E before I, whatever. It is the full range of proteins coded for by the genome of an organism. There you go. So it's all the proteins that can be coded for by an organism or by an organism's DNA or by its genome. 7.2, give two structural differences between a molecule of messenger RNA called mRNA and a molecule of transfer RNA, which we call tRNA. So hopefully mRNA is going to kind of looks like that, and tRNA is the one with the oh my good gosh, it's got the like clover shape, right? I'm sure I've done that really badly. And it has that binding site which it attaches amino acids to, and then it has that anticodon. So I could have probably drawn that up there as it attaches there. Okay, let's put this down then. So there's a million answers you could do here. mRNA has more nucleotides, as really that could have like 1,000 nucleotides, in the primary structure of the polypeptide it's coding for, whereas the tRNA has just got the anticodon, which is what? That's made up of three bases, so three nucleotides involved there. So mRNA has more nucleotides, should get us a mark. Um, mRNA is linear, whereas tRNA in a clover shape, so it's not in a linear shape. It's folded round by hydrogen bonding, which you don't get in mRNA. We could say tRNA contains anticodon, whereas mRNA has the codon. Oh, tRNA. Oh my gosh, tRNA has an amino acid, and this comes up quite a bit actually, amino acid binding site, whereas mRNA does not. I reckon that's enough there, guys. You only needed two of these, but I just put in a few there. You can check the mark scheme. You'll find that on the Physics and Maths Tutor website or on the AQA website, and you can check the mark scheme for any extras. There's probably loads of extras between them, other ones between them. Yeah, okay. Lots of similarities as well, which they haven't asked us for. Okay, 7.3, starting with mRNA in the cytoplasm, describe how translation leads to the production of a poly. Basically, 5 marks, talk about translation. Do not include descriptions of transcription and splicing. So they don't want us to say, oh, the mRNA leaves via a nuclear pore. They don't want us to say it's pre-mRNA. They don't want us to say that the introns are removed by splicing. They want us to go from after splicing. So what I want to say is, I'm going to start like this. mRNA attaches to ribosome. Then I'm going to say tRNA brings an amino acid to, oh my days, mRNA. Then I want to say we're basically talking about translation here, and translation happens later. It's a little cool way to remember that it happens after transcription, translation later. tRNA has anticodon, binds to codon on mRNA. We want to say by comp base pairing. You can't write comp. I can because I'm trying to speed it out. Right? So complementary base pairing. We've brought the amino acid down, bronzed the amino acid. Check out the spelling. So mRNA attaches to ribosome, brings amino acid to the mRNA. tRNA has an anticodon, binds to the codon on mRNA by comp base pairing. What can we say next? Oh, a secondond, a second tRNA brings another amino acid. This binds to the first by a condensation reaction, forming a polypeptide. No, forming a peptide bond. Oh my gosh. Oh my gosh. The spelling is the typing. I've got fat fingers, I'm afraid. Put on a bit of weight. My fingers are so fat now, they cover about 6 keys on the keyboard every time I try to type. You should see me on a mobile phone. If I try and text, I get about half the alphabet coming out with each press. Right, this binds to the first by a condensation reaction, forming a peptide. And this carries on until oh my days, until a stop codon is reached. Where the polypeptide leaves the ribosome. Oh my gosh, I'm giving up with the spelling now. I'm giving up, giving up, giving up. Okay, there's probably some other things I could mention there. For instance, I talked about carries on until a stop codon. I should have said this process carries on until a stop codon is reached or is repeated with many tRNAs bringing. I could say the previous tRNA always leaves after the peptide bond is formed. And I could have mentioned the start codon, which would have been kind of better to the flow, right? I could have said tRNA brings an amino acid to the mRNA at the start codon. So I could have mentioned start codon there as well to get maximize my marking. Anyway, that's the end of question 7. Let's go. All right, folks, we're on question 8. So 8.1, state and explain the property of water that can help buffer changes in temperature. So what does this mean, buffer? What it means by buffer is prevent the temperature from going too high or too low. So what is it? We need to pay attention to the command words here. So we're going to get 2 marks. We're getting 1 mark for stating the property of water that allows it to buffer changes in temperature. And then the second mark is to explain how it does that, basically, so to speak. Okay, it's only 2 marks, so we don't need to go into too much detail. Well, of course, the property is water has a relatively, you don't have to say relatively, but it has a relatively high specific heat capacity. So when you compare it to really what it should have, it has a high specific heat capacity. Now, why does it have a high specific heat capacity? Well, water is a polar molecule, and hydrogen bonds can form between adjacent water molecules, and hydrogen bonds require more energy than other weaker intermolecular forces, and this gives water a high specific heat capacity. Now, we don't need to talk all about any of that in our explanation. We just need to say what having a high specific heat capacity means. And so what does this mean? That's what they mean by explain. State the property, the high specific heat capacity. Now can you explain what it means? This means it requires a lot of energy to raise the temperature of water. So water temperature remains relatively stable. And this is the reason why water is such an incredible substance on the earth. It's the reason why life probably evolved from the water. It's the most stable environment on the earth. The air is not so good, obviously. The atmosphere, the air, that changes temperature really easily and varies throughout the day, whereas the water temperature has a much smaller range of fluctuation. So it's a much more stable environment without any massive changes. And that is because it requires a lot of energy to raise the temperature of water, or water has to lose a lot of energy for the water to temperature to decrease. So it works both ways. And there we go. You only need 2 points for this. So this first point is you have to get that, a high specific heat capacity. And then these are really the explanation is here. It requires a lot of energy to change temperature of water. Okay, enough of my whiffle waffle. Let's move on. 8.2, water is used to hydrolyze ATP. When you see the word LYSE, lysis, it means breaking. Hydro means with water. So water is used to hydrolyze ATP. Name two products of ATP hydrolysis. You know this. You should be able to write this down. If you don't know this, you need to give yourself a slap in the face, because this is some basic stuff that you're going to need time and time again throughout your A-level biology. So learn this off by heart. And of course, our products are ADP, adenosine diphosphate, or an inorganic phosphate. So I'm just going to put ADP and PI because I'm lazy. So adenosine diphosphate, and we've got inorganic phosphate, which is represented by the PI symbol, and that's going to get us our 2 little marks. Okay, what am I ticking for? Jeez. Let's move on. Oh, great. We love a fat question with a big stem. No, Diddy. This is called the stem of the question. So hydrolysis of ATP is catalyzed by the enzyme ATP hydrolase. A student investigated the effect of ATP concentration on the activity of ATP hydrolase. She used shortening strips of muscle tissue caused by contraction as the evidence that ATP has been hydrolyzed. So she's trying to show that when you hydrolyze ATP, the muscle gets shorter, and this is evidence for ATP activity. She took 4 slides A, B, C, and D. So we've got slide A, B, C, and D in our table here, and added strips of muscle tissue of the same length. So each of the muscle tissues that were added to A, B, C, and D were the same length. She then added the same volume. So she's using the same volume. That's a good control variable of ATP solutions of different concentrations. Obviously, independent variable, she's changing the concentration in mole per decimeter. What else? What else? What else? Okay, she left them for 5 minutes. Then she recorded the final length of each strip. So what is the effect of ATP on the length of muscle tissue? They'll probably ask us that, but you should be bearing that in mind as you skim read through this. Other than those given, name 2 variables the student could have controlled or should have controlled. Well, we can see that she already controlled volume. So let's not say volume because she controlled it. She was using different concentrations, so that's not a control variable. She measured the initial length and final length. That was her dependent variable, so we can't say she's going to control the final length. So what could she control? Well, we've got this solution in moles per decimeter. She changed that. Concentration, we can't say to change that. We can't say volume. Final. What else about the muscle tissue? Now, she says she's controlled or she was measuring the length, and she used the same length to each slide, the same initial length. I think she needs to keep the thickness or the width the same. So let's put thickness of muscle tissue. That would be sensible. The thickness of the muscle tissue. How long does she leave it for? 5 minutes. So we don't need that. What else? Let's have a little think. Let's have a think. What else could we go for? So we could also look at where the muscle tissue came from. Is she using human muscle tissue, rat muscle tissue, frog muscle tissue, or a mixture? So maybe say same species. Oh my gosh, same species um, of animal providing muscle tissue. I mean, you could get into the age of the animal that's providing the muscle tissue and all of that as well. We could also talk about factors that may affect the rate or the speed at which ATP um, breaks down and releases that ADP and PI, which would be dependent on temperature. So maybe the temperature of the muscle tissue would be another good one, or the temperature of the ATP solution. Okay, let's move on. 8.4, describe and explain the pattern shown by the data in table 1. So my description would be as the concentration of ATP solution increases, the final length of muscle tissue decreases. So that is my describe. With describe, don't use any science, just say what the table shows you. So as the ATP concentration, I should say solution, as the ATP concentration, but I'm being quick, increases, the length of muscle tissue decreases. Now, what's the explanation? Why? What makes a muscle shorten in length? And of course, it's that the muscle is contracting. So the more ATP, the more um, energy can be released for muscle contraction. And you won't have done it yet because you're doing AS, but yeah, there's something called the power stroke. The actual contraction of the muscle requires the breakdown of an ATP molecule. Also, to remove the myosin heads from the actin binding sites requires ATP as well. So there's two areas that require ATP. Right, let's zing down. We've got another part to this question. 8.5. The hydrolysis of 1 decimeter of 1 mole per decimeter solution of ATP releases 30,500 joules of energy. 60% of the energy released during the hydrolysis of 1 mole per decimeter of ATP is released as heat. The rest is used for muscle contraction. The student added 0.05 centimeter cubed of ATP solution to slide D. Calculate the energy available from ATP for contraction of the muscle on this slide. Right, I did this question and got it wrong the first time I did this question through. I remember this years ago in class teaching this and messing it up, on all from a very simple mistake. So I'm going to go through and show you how to do it, and I'm going to include my little mistake I made because some of you might make the mistake too. Why don't you grab a pen and paper, pause the video, have a go, and then unpause and watch the explanation. Okay, so what we're saying is that if you have 1 decimeter, now a decimeter is 1,000 centimeters. You might say, hey, why are we converting to 1,000 centimeter cubed? Well, we're converting to 1,000 centimeters because here we're using centimeters. The student added 0.05. So I need to make my units of volume the same. So I could turn that into decimeters, or I could turn this into centimeters. I'm going for that into centimeters. So hydrolysis of 1,000 centimeter cubed of 1 mole per decimeter solution releases 30,500 joules. It's only 60% wasted as heat. So 60% wasted as heat. So if you want, we can 60% wasted. So 40% is used in contraction. Don't know why I'm drawing these arrows all over the place. So 40% must be used in contraction. So let's find out how much energy is used in contraction. And so I'm going to do 0.4 times 30,500, and that is going to give me 12,200 joules. So what I'm working with here is 12,200 joules are actually used in contraction. And I think if you put that there, you might get yourself one of the 3 marks. Now, I want to know, bada bada bing, bada bada bang, bada bada bing, bada bang, bada bong. I want to know how many, yeah, how much energy then, that is for 1,000 centimeters. So how much energy will 0.05 do? So you could do it this way. There's a number of ways to do it. So you could do 0.05 divided by 1,000 times the 40% of the energy of 1 mole. So we'll try that. Let's do that times 0.05 divided by 1,000. And that is going to give you as your answer 0.61. And the mark scheme will give you maybe 1 or 2 marks for that, but not all the marks. And the reason is, so you've done everything right. If you've got this far, and it doesn't matter, you could do 1,000 divided by 0.05. And if you do that, you get 20,000. And then you go 12,200 divided by 20,000, and that would get you 0.61. So there's a number of different ways to do this. We could have left that as 30,500, and we could have done 30,500 times 0.05 times 1,000, and you get like, I don't know, 1.25 or 1.525, something like that. And then you can times that by 40% and get 0.61. So it'll be like 1.25, whatever it is. And so that's another way to do it, but you're going to end up with this 0.61. And for a while, I was like, oh my gosh, why aren't I getting the right answer? Because this looks like it should be 0.61 joules per centimeter. But that's assuming that we're using a 1 mole per decimeter solution of ATP. And look what they've put in bold. So my big warning here is anytime they put something in bold, i.e. dark black, they are telling you, pay attention. This is really important. Go check it out. So what we're going to do, we're going to zing up here to the table. Oh, let me get rid of that off the screeny. And let's check the table out. So what it shows us here is that slide D, the concentration of ATP solution is 8 times 10 to the minus 6. So we've got 8 times 10 to the minus 6. Yeah, so that is the concentration. So what we need to do, oh my gosh, yes, is go back down and multiply our answer by the concentration. So let's do that. Yeah, basically, what you've probably done if you've flopped on this question is you just haven't taken into account, I can't get that to line up. Oh my days, the concentration. So it's times 8 times 10 to the minus 6. Let's see what we get when we do that. 0.61 multiplied by 8 times 10 to the minus 6. And we get the correct answer, which is 4.88 times 10 to the minus 6 joules. Sorry, I just let my dog out. I can hear it messing around in the garden. Okay, so there we go. Personally, I'd give us a mark for, what would they give us a mark for? Well, you'd have to check the mark scheme. It's 3 fat marks. So they're normally easy to get 1 mark. So probably if any evidence of 12,000, 1,200 means you've done the 40% bit. 0.05 over 1,000 times that. 0.61 is going to be a mark. And then there's a mark. And also, I think if you did it without doing the 40% first, you might get a mark as well. So, okay, let's zing. This looks nice and easy. 9.1, describe the pathway taken by an oxygen molecule from an alveolus to the blood. So we're going from the path of an oxygen molecule from the alveolus to the blood. So that sounds pretty easy, right? So what have we got here? We've got the alveolus, which has a thin wall, 1 cell thick, made of epithelial cells. We got the oxygen there. So the path is it's going to diffuse through the wall of the alveolus, then through the wall of the capillary, and then across plasma into the red blood cell. So that's what I'm going with. Describe the pathway of an oxygen molecule. Yeah, from an alveolus to the blood. Okay, so I want to say diffuses across alveoli wall, then across capillary wall, across plasma and into a red blood cell. Whether we have to say across the membrane of a red blood cell, it's probably advisable not to take any chances because you just don't know until you look at the mark scheme, you just don't know which of these are on the mark scheme. It'd be something like the word diffusion is 1 mark and then across the capillary wall and you're like, oh, easy peasy. Well, you might as well put across the plasma into the red blood cell, across the red blood cell membrane, and then attaches to the hemoglobin, if you like, becomes oxyhemoglobin. You know, they're not going to deduct marks. They'll just say ignore if they're like not interested in it. Now it says explain how one feature of the alveolus allows for efficient gas exchange to occur. So you've got to state the feature that gets you 1 mark. So I'm going to say thin walls. We probably have to say 1 cell thick. What does this do? This creates a short diffusion pathway. Now, there you go. That is the adaptation and the explanation. Something else, we could say large blood supply. Um, basically it has a large capillary network around each alveoli is a large capillary. What the hell's that network? Right. And what does that do? That maintains a steep concentration gradient. Um, let's add in diffusion, eh? Let's add in diffusion. A steep concentration diffusion gradient. Oh God, whatever. Um, from the alveoli to the blood. There we go. So you're going to get a rapid rate of diffusion because of that. So you're going to get faster. And maybe I need to also say, but any of these two explanations they result in a higher rate of diffusion. And that would just make sure I cover everything. Of course, the alveoli is folded, so it has a large surface area. This will increase the rate of diffusion too. All right, let's zing on. We don't want to be here all night. Oh my days. Carbon monoxide is a poisonous gas that's present in cigarette smoke. This carbon monoxide can be absorbed into the blood where it binds with hemoglobin. Scientists investigated the concentration of carbon monoxide in cars in which people were smoking or not smoking. They measured the concentration with the car windows open and closed. Figure 7 shows the results as they presented them. A value of plus or minus 2 standard deviations from the mean includes over 95% of the data. And that means they're telling you that these error bars are decent because they are 95%. So we got the mean concentration of carbon monoxide inside in the air inside the car in parts per million. So when people weren't smoking with the window open, um, there was more carbon dioxide inside the car than with the window closed. And then when people smoked for 5 minutes, closing the window meant there was more carbon monoxide inside the car. And then 5 minutes after smoking, um, there was a significantly higher amount of carbon monoxide in the car than outside. Okay, what do we want to do? Or when the window was open, not outside, sorry. Blimey. In England in October 2015, a law was introduced making it illegal to smoke in a car carrying someone who's under age 18. Following the introduction of the law, a politician stated, it is dangerous to smoke when a child is in the car. Higher levels of deadly toxins can build up, even on short journeys, and children breathe faster than adults, meaning they inhale more of the deadly toxins. Use the information provided and the data in Figure 7 to evaluate the politician's statements. Well, this is going to be a little bit difficult, isn't it? Their statement has no relation to what we're just looking at. So there's loads of things to criticize about using this data. Um, there's loads of things which you would criticize about this statement if you were trying to use this data. So carbon monoxide is dangerous, binds to hemoglobin, prevents the formation of oxyhemoglobin. It is a toxic substance, carbon monoxide. Okay, so it is a dangerous toxic substance. And what we would say is, yep, even on short journeys and children breathe faster. Okay, so positive thing, carbon monoxide is dangerous. Well, not positive, really. We would say, yeah, short journeys. They did journeys not shorter. They only did journeys of, well, this data is only relevant for journeys that are at least 5 minutes. And it shows that if you've been smoking for 5 minutes, there is a higher carbon monoxide concentration, and it is a significant amount with the window closed than if you weren't smoking. However, if you had been smoking for 5 minutes, we can see that, and the window is open. This is a however, there is no significant difference between not smoking and smoking with the window open on a 5 minute journey. So actually, the politician may be wrong there because actually the carbon monoxide level is not significantly different. I'm just looking at the two white bars here, guys. Look at this white bar and this white bar. This is smoking for 5 minutes with window open. This is not smoking and the window open. And you can see there's overlap between the bars. So there's no significant difference in the amount of carbon monoxide inside the car when you're smoking for 5 minutes with the window open as to when Uh, you're not smoking with the window open. Very, very similar. Now we can see here that 5 minutes after smoking inside the car, when the window's open, the carbon monoxide levels are exactly the same as, yeah, there's no significant difference. Again, they're actually exactly the same as when not smoking with the window open. But we can, so what we can conclude is that smoking with the window closed, even for just 5 minutes, there is a significant difference in the amount of carbon monoxide inside the car. So, yes, this would be more dangerous to young people. Now, other things to take away from this evaluation is that the statement talked about toxins. It did not say relate specifically to carbon monoxide, and our data is only on carbon monoxide. So what we would say there is, um, that's not so good. Our data is only on carbon monoxide. We cannot back up the rest of the politician's statement about other toxins, the many other toxins found in cigarette smoke. Um, what else? What else? They didn't say how many people did the study. They didn't say how many cars they tested. We don't actually know that kind of stuff. So we don't know how big the sample size is. We don't know if it was representative. We don't know the make of the car. We don't know anything else about it. Lots of other factors we don't know. So the kind of things I would go on about here, I'd be let's write this all up, yeah, so you can see the answer. Okay, so here's a bit of an answer you could go with. So no significant difference in the CO levels when the window is open. Um, yeah, and that was when smoking or not smoking. So that's kind of like against this. However, after 5 minutes with the window closed, there is a significant difference in the CO levels, which is dangerous as CO is a toxin. We don't know the rate at which the child is breathing, so that's an issue with this study. Like how far, what is the child's breathing rate? And we also, children have smaller lungs. So although they might breathe faster than adults, because they have smaller lungs, they can only take in so much air. So they may be taking in less CO with each breath. And it may work out that the total amount of carbon monoxide taken in between an adult and a child is the same because the child's breathing faster but can't take in as much air with each breath, whereas the adult is breathing slower but taking in a greater volume of air. And here we go. There's plenty of other points you could make here that aren't on the mark scheme that should be accepted because they would be relevant in an evaluation of this. Okay, this is an AS Biology question 10, quite a difficult one actually, because this will help you prepare for your A-level biology paper 2, the final question, because it's basically a comprehension question, which is what you get at the end of A-level paper 2 when you do follow the biology all the way through. But this is AS, so anyway, good practice for that. Comprehension, read the following passage. So we're going to read this passage, then we're going to answer questions specifically about the passage. So read the following passage. Sizes of populations of normal intestinal bacteria are usually controlled by T cells that are produced slowly and in small numbers by the immune system. So if you wanted to highlight, I suggest you'd underline sort of they're produced slowly and in small numbers normally. These T cells do not normally survive for long, so they're short-lived. As a result, they do not release large amounts of cytokines. So I'd underline cytokines. Cytokines are chemicals that can cause the swelling of the lining of the intestines. Of course, cytokines are used for cell signaling in the immune response. Crohn's disease is a long-lasting disease that causes swelling of the lining of the intestines. It's believed that Crohn's disease can be caused by a loss of tolerance to normal intestinal bacteria, as shown by an unusually large response by T cells. This response can be triggered by pathogenic bacteria in the intestines of people with a genetic tendency to Crohn's disease. Some people's Crohn's disease can be controlled by a drug called 5-aminosalicylic acid, or 5-ASA, that reduces swelling. Another drug called 6-mercaptopurine, on my days, otherwise known as 6-MP, may also be used. 6-MP inhibits an enzyme required to make adenine and guanine. This is effective because most cells can recycle nucleotides, but T cells are not able to do so. That means that when T cells want to reproduce and make more of themselves, they have to have that enzyme in order to make the adenine and the guanine. They can't recycle old adenine and guanine from other cells. Use information from the passage and your own knowledge to answer the questions. All right, let's get on and see what the questions are. So, question 1, worth 3 marks, 10.1. The Crohn's disease symptom of swelling of the lining of the intestines could be triggered by pathogenic bacteria in the intestines. Lines 6 and 10, Let's suggest how. So they're saying that pathogenic bacteria in the intestines could trigger the Crohn's disease symptom. And it says lines 6 to 10. So we're specifically going to read lines 6 to 10. Crohn's disease is a long-lasting disease that causes swelling of the lining of the intestines. It's believed that Crohn's disease is caused by a loss of tolerance to normal intestinal bacteria, as shown by an unusually large response by T cells. This response can be triggered by pathogenic bacteria in the intestines of people with a genetic tendency to Crohn's disease. So what they're saying here is that the bacteria, the pathogenic bacteria, appear in our intestines, and essentially our T cells. Remember, T cells have a receptor. Here's my T helper cell or whatever T. It's my THC. They have a receptor. So once it binds to the pathogenic bacteria, it then uses cytokines, which are the cell signaling molecule of the immune system. That it's a big family name for a whole group of different cell signaling molecules. But the cytokines are released and they stimulate T cells to divide, and you end up with lots more T cells. So this question is saying, how is it that the presence of pathogenic bacteria can cause the symptoms of Crohn's disease? So we want to basically put that into words and say the T cells have a protein receptor that binds to the specific antigen on the pathogen. Therefore, the T cells clone themselves, and we get many, many T cells produced. The cytokines, because it says people with Crohn's disease, where did it mention? Cytokines are chemicals that can cause swelling of the lining. So there's lots of cytokines about which cause swelling of the lining of the intestines. So that's one of the reasons, is that the T cells are cloning themselves in response to the pathogenic bacteria, causing swelling of the intestines because of the amount of cytokines, increased amount of cytokines. But also, the T cells normally are short-lived. So maybe these T cells, normally they don't survive for very long. Maybe these T cells survive for a long time. That's part of the presence with Crohn's disease. And also, maybe these T cells are also responding to the normal bacteria, which they're also um, binding to the normal bacteria and producing T cells in response to that. And we've just got a load of cytokines and swelling and all of that business. And we just need to get that down for, what was it, 3 marks. So let's go and put that down. All right. That was some crusty drawing, but that's my level 3 GCSE art, although back in the day it was called a D grade. Okay, so what do we want to say? We want to say, okay, so here's the answer I've gone for. Pathogenic bacteria have an antigen, right, that bonds. Oh my God, that's supposed to say binds. What an idiot. That binds to T helper cell receptor. So the T helper cells bind to the pathogenic bacteria's antigen, and they stimulate a large T cell response. They might also respond to the normal bacteria's antigens. Once they're triggered by the pathogenic bacteria, they're also recognizing the normal bacteria as foreign. And so we get this really large response. Now, what T cells release when T helper cells are activated, they release cytokines. So we're having a large release of cytokines. We were told in the passage before line 6 to 10 that cytokines cause swelling of the intestines. So that's going to get us a mark. When T cells divide, they make memory T cells, killer T cells. The mark scheme doesn't specifically say this, but this is just for your own understanding. So these T cells are using cytokines to stimulate other T cells to clone themselves and differentiate into memory T cells and killer T cells. And another suggestion on the mark scheme is that these T cells live longer than normal. They're supposed to be short-lived, but there's an issue where these T cells live longer than normal. Therefore, they're constantly producing cytokines that are causing the intestines to swell. So the marks really are for saying that the antigen is recognized by the T helper cell. That the T helper cells also respond to normal bacteria is a suggestion that will get you a mark. That they release cytokines and that there's an increase in the amount of cytokines causing the intestines to swell, and that they live longer than normal. So all of these, you only needed 3 of these 5 to get a mark. All right, quite a hard question, I think. Let's zing to the next question. So the next question says, suggest the meaning of a genetic tendency to Crohn's disease, line 10. So why would some people have a genetic tendency to Crohn's disease? And this would be down to they have a genetic mutation. So they've got a mutation, a change in one of their nucleotide base sequences that means they make a protein or they have an allele. So they have a genetic mutation which has given rise to an allele, um, which is a type of gene. So they've got a genetic mutation which has given rise to an allele, a type of gene, basically. You could say gene here. That means they have a larger T cell immune response, or maybe that they produce more cytokines than normal, causing more swelling of the intestines. Or maybe it's that if you have this allele, your T cells recognize normal bacteria as foreign too. That causes T cells to respond to the antigen on normal bacteria too. So normally your T cells would not respond. They've evolved to not respond to the normal bacteria. We live in a symbiotic relationship or mutualistic relationship with those bacteria. Oh my days, look at that word antigen. Oh, what that is a funky way to spell it. Antigen. Let's put that properly. Antigen. Yeah, you get the idea. Cool. Any two of these. So they have a gene, which means they get a large T cell response, or that they recognize the normal bacteria as foreign too. Okay, let's move on. Suggest why 5-ASA, that was that like 5-amino salicylic acid, is only effective in controlling the swelling of the lining of the intestines in some people with Crohn's disease. So why does this only work on some people who have Crohn's disease? So what we want to do here is go up and check out lines, what did it say, 11 and 12. So it's about here. Some people with Crohn's disease, some people's Crohn's, so not everyone with Crohn's disease, can be controlled by a drug called 5-amino salicylic acid that reduces swelling of the intestines. Okay, so the salicylic acid reduces swelling of the intestines. Why might not that work for everyone with Crohn's? Well, they might just produce too much cytokines. So some people produce so much cytokines, I should say a larger volume of cytokines, or yeah, you get the idea, that I'm just going to call it the drug cannot reduce the swelling enough. And then, of course, down to some people's biology, some people may have side effects or the drug doesn't work in some people. For some people, they may be allergic to the drug. Instead of saying drug, I would use the 5SA, but I couldn't remember the name. So what was it? 5-ASA. Oh, 5-ASA is what it is. Suggest why 5-ASA is only effective. Oh, I've just answered that in the wrong place. Sorry, folks. Never mind. Moving on. It's all falling apart here. Right, 10.4. We're near the end of the paper and I'm knackered. Suggest why 6-MP. I'm not even going to pronounce that one. That was really hard. Mecatoporin, whatever it was, can be used to control the symptoms of Crohn's disease, lines 13 to 15. Do not include details of enzyme inhibition or protein synthesis. Thanks for that. So guanine. So cannot reproduce. So they can't make new nucleotides of guanine. And what was it? Adenine. I think it was guanine and adenine. So cannot reproduce. Therefore, we do not get a large volume of cytokines produced. So the lining of the intestines does not swell as much. Surely that's got to be 3 marks. What else do I need to add? Oh, we need to get a little bit more A-level here and specific, I reckon. So the T cells cannot make new nucleotides of guanine and adenine and cannot recycle like other cells. Other cells can recycle the nucleotides, so cannot reproduce as they cannot do DNA replication. So they can't do the semi-conservative replication of DNA because they don't have any free nucleotides of guanine and adenine, and they're unable to recycle other nucleotides. So therefore, we do not get a large volume of cytokines produced, so the lining of the intestines doesn't swell as much. Boom, 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 bang. Right. Okay. That's the end of the test, people. Don't forget to share, like, and subscribe. Helps out the channel. Check out all the other AS Biology paper ones I've done. I think I've just got the 2020 paper to do, which will be coming shortly to you. And once we hit August, I'll be releasing the 2024 AS Biology paper one. All right, folks, that's us done for today. So, you know the coup. Sharing's caring. Hit the like, punch up the bell, subscribe, all that jazz. Let's get to the marks.
You're still here? It's over.